I close my eyes and let my heart go back in time, you're still here like you were yesterday. I can make your heart stand still.
So, in continuation to the first session of the lecture, now let's try to define genes from the perspective of the modern molecular genetics point of view. So in the classical genetics, the genes was viewed as a lab factor that defines the characters. But in reality, it turns out that the genes are actually some area of the DNA. So the segment of DNA. Yeah. So not all, but only some portion of the DNA that carries the information for protein synthesis. But that has been the earlier but narrow definition of the genes. So in associated with this, really, one gene is one protein hypothesis. It's actually rather very famous hypothesis, which was actually later on proven experimentally, like in early 1970s and something like that. So at the time, the gene was viewed as some area of the DNA that produced the protein. So each gene is responsible for making one protein, which is it is a right concept. But if we only stick with this one gene, one protein hypothesis, by the way, this one gene, one protein rule only applies to the case of eukaryote. The prokaryotes, their gene organization is something a little bit interesting. So we cannot really say the prokaryotic gene is applies to one gene and one protein hypothesis always. But let's forget about that. Any deviation from standard, there is no reason why we dig into any unnecessary confusion and complexity. So let's stick with this. So one gene and one protein hypothesis. What's wrong with that? First off, here under the one gene one protein hypothesis means, there is a one gene and another gene, then three gene. Although in this description there is no intergenic region, but in reality, of course, there are many. These genes should be separated by huge area of non-gene and intergenic regions. But anyway, okay, so if we only zoom in, look at the genes, the third gene, gene 3, which is the nothing but the DNA sequence. So DNA sequence, like whatever, AAA, CC, GG, it goes like that. So this is a part of this gene 3, and this gene will be producing RNA, which is complementary to this AU. You see the relationship between the DNA and RNA. So that's why RNA can be referred as a copy of DNA. But then this RNA, the information present in RNA, which we are going to take a look at in a little bit more detail later on this chapter, organized in specific ways. So 3 nucleotide by 3 nucleotide forming a single unit, and each 3 nucleotide will specify a specific amino acid. So that's how the information present in the RNA can be converted into actual the nature identity of amino acids to produce a protein. Here polypeptide means simply a protein. I have already went over this distinction between whether when a polypeptide can be taken as a synonymous to protein, and when others, in some other occasions, polypeptide is different from a protein, but that's not any important. So that's the definition of a narrow definition of gene in molecular genetics. But you have a problem, and under such definitions, some other gene products are excluded. That's not fair for them. So here, a broader definition of the gene can be said as the segment of the DNA, but not only making protein, but any area of the DNA that potentially you can be on vacation for the moment, but as long as you have a potential of making RNA, then you are a legitimate gene, is the definition of this the broader definition of DNA. So any area of the DNA that is capable of generating the RNA, then they are the gene. So in other words, the segment of the DNA that is capable of the activity of a transcription is also kind of a broad definition of the gene. So at this point, probably although I have introduced 3 different types of RNA earlier in the class, but now let's become a little bit more serious, so that actually how many different types of RNAs do exist in these days actually. But here the reason why I try to bring up different kinds of RNA is only this: the distinction between coding RNA versus non-coding RNA. Coding RNA means the RNA that the information, DNA, the sequence of RNA directly converted into the protein, which means the amino acid. Like I showed 2 slides ago, 3 RNA nucleotide sequence acting as one single unit, and each of which signify a specific amino acid. That is an example of coding RNA. So those RNA sequence that is used in signifying each amino acid in protein building is a coding RNA. On the other hand, non-coding RNAs are those RNAs that not directly involve, that the RNA sequences are irrelevant in terms of there is no corresponding amino acid sequence for this non-coding RNA, so they are not used directly in protein building process. But some of them are, at least some of them, most of them, should I say, some of them are indirectly related involved in protein synthesis, and without it protein synthesis cannot occur. So that's the difference between coding and non-coding RNA. So having with that in mind, okay, so let's take a look at different kinds of RNA here. So different kinds of RNAs are made. as so all these RNAs are, whether what kind of RNAs, but they are all in nature, all of them are complementary copies of DNA, which means like we have seen, like, if DNA sequence was like GGTAC like that, then RNA sequence which is made is UUUCC AUG, like, okay. So this is the RNA, but then we know the DNA exists in double helical, double strand, so only one of the two strands will be used in such copying, I mean complementary. Copying. So in RNA synthesis, what they do is simply pick up any RNA nucleotide which is complementary to this particular area of DNA sequence. So this DNA sequence providing that provides the information for this copying procedure is called template DNA. And obviously we all know that there is another DNA sequence which is complementary to this template DNA, and actually we will get to see later on, but the template DNA, the other complementary DNA which is non-template DNA, would be like in nature, its nature would be TTT CC A T G. So this is non-template DNA, but if you compare with this, wow, this is exactly identical to the RNA sequences, but simply T to U conversion, because thymine is not present in RNA. This, so except that RNA sequence, so that the copying, the template DNA would be identical to the other strand of this same area of the DNA, template DNA, which obviously understandable because that's the nature of this DNA double strand, which is complementary. And if RNA is also being made through this complementarity, then what else can you expect? So anyway, having said that, all those RNAs are complementary copies of the template DNA, and there is messenger RNA, that famous messenger RNA. It is a coding RNA, and actually this is the only coding RNA among these 6 different kinds of RNA that we are going to see in right over here. And its job is now the messenger RNA sequence will specify amino acids as a unit of a codon. One unit is codon, is a 3 nucleotide. 3 RNA nucleotide, of course, yes. Another kind of RNA is a ribosomal RNA, we have already seen. And ribosomal RNA, since it is not involved in protein coding, so it is a non-coding RNA. But without it, because you cannot assemble the ribosome, without ribosome you cannot make this protein synthesis get going. So, and also more importantly, or maybe less importantly, okay, but as equally important, some ribosomal RNA actually is the one making this peptide bond, which is responsible for connecting each amino acid in protein synthesis, but also some other ribosomal RNA can also act as other types of ribozyme too. So that means it can generate some unique three-dimensional structures, as you can see over here. This is one such type of ribosomal RNA, 16S-RNA, which is a prokaryotic 16S-RNA. But as you can see here, there are so many secondary structure which are the result of all intra-strand complementary base pair. And if you have this intra-strand complementary base pair, here this is only the secondary structure, but overall in 3D structure it will be in an existence of a tertiary, three-dimensional tertiary structure as well, which is the basis for unique shape, means unique function. So that's why some of them can act as enzyme like protein. And transfer RNA is also non-coding RNA, and its function is a carrier of the individual amino acid into the ribosome so that each individual amino acid can be assembled together. So this is absolutely a necessary component for protein building, the translation procedure too. And this is the structure of typical transfer RNA in this area, which is 3' end of this RNA. You know what the 3' and the 5' end, right? The 5' end is the first nucleotide of this whole polymer of a nucleic acid as an RNA strand, and this is 5' and then the last part is a 3' end where different amino acids can be attached. So since we have 20 different amino acids, which means at least we should have 20 different transfer RNA, each of which can recognize and then deliver after this hooking up to the ribosome. And the secondary structure formation based intra-strand complementary base pair also can you see over here, and as a result this will exist in the unique 3D structure like that here. So secondary structure and tertiary structure, which is model is like their usual way of existence like that. So this is exactly what I have just said, 20 different. But how many different 20 the transfer RNA? It's more than 20, even though there are only 20 different amino acids that become a part of this protein building process. But the number of different transfer RNA in the cell far exceed the 20, which appear to be the minimum number that is required for handling this amino acids carrying business. We will get to that later in this chapter. And so up until then, those 3 RNAs I have already mentioned earlier, but this is a new mentioning. Well, actually this was the one discovered relatively later on. This is called microRNA and abbreviated in the mRNA. MicroRNA is a kind of a new kids on the block, so to speak, and its existence only became known to the scientific community I guess it's only like early 1990s, so it's relatively very new discovery. And it is a non-coding RNA too. Yes, it is non-coding RNA, but it has a very interesting feature as a non-coding RNA. But first off, what does it do? It is involved in gene expression control over certain genes. To better understand this gene expression control, probably you better understand what's this gene expression, what does it take to have any control in gene expression. That's another part of the purpose of this chapter, to understand the nature and mechanism of this gene expression is. So obviously we will get to that later, somewhere in this chapter. And interestingly, although it is not a coding RNA, but it has a very similar in terms of its sequences, complement to specific target genes, which is almost as is very similar to messenger RNA. But this messenger RNA and the microRNA are two different things. If I briefly try to distinguish the microRNA from messenger RNA, is that okay? If this was the DNA sequence, for example, GAA T C A G A C, for example. Let's say this is or gene, and if this gene make messenger RNA, and it should be a complementary base pair of, so it should be C U A A G U C U G B is messenger RNA. Okay. Fine. However, in the case of if this area of the gene also provide, it doesn't actually, no, the same gene does not provide both messenger RNA and microRNA, so that's wrong. However, in the genome there is so this is one gene, and somewhere else elsewhere there is a gene sequence exists, like, which is very similar but not identical to this gene A. Let's say this is gene A. And what I mean by that is a G A C T, for example, A C A C A C. This is a microRNA, which is its sequence is very similar to A but not identical. So here you have this sequence and this sequence. This sequence is different from this DNA sequence originally. So that's an example of what's microRNA, and that's the feature actually acting importantly in terms of its playing its function as the gene expression controller. So this microRNA will control the expression of gene A. Really? Yeah, after this microRNA DNA generate the RNA sequence which is complementary to this, so should be CUGA UG UG UG, because this one will have a complementary base pairing with this messenger RNA produced, although there are some mismatches. Here some mismatches, CU here mismatches, AU mismatch, and GU, G, C, C. Okay, here is a mismatch. But overall, these two microRNA can complementary pair with a messenger RNA of similar sequence, because a little bit of the mismatches can be tolerated during. So this complementary pair means, another way of describing this complementary pair is called hybridize. And that's the basis of this. So it has a sequence complementarity to specific target genes, and that target gene, by virtue of that specific target gene, the expression is controlled by the microRNA is the one. So that's the description of. So there is a gene, and somewhere else the microRNA target sites, which is very similar. Oh no, this gene has some portion which is very similar to a specific microRNA. So this we call as a microRNA target site, and then actual microRNA will hybridize over here, and that invites some of RNA degrading machinery, and this whole RNA will be degraded, or I don't want to really go into any complex details, but anyway, whole production, generally the production of this messenger RNA production will be inhibited. That's the role of this particular microRNA. And it turns out that its role is actually very broad and complex, so many different variety of functions, and very important. If not every genes, like in human, almost 23 different genes, every different gene has its own partnering microRNA. I'm not saying that is the case, but at least some of the genes, for some of the genes, actually there is a specific corresponding microRNAs genes are present in the genome, and the expression of such microRNA will be able to hybridize the the gene product of that particular target gene, the RNA, messenger RNA versus microRNA, and once they have this hybridization, then the product of the protein out of those messenger RNA will be specifically inhibited. That's what I'm saying, the gene expression control. Okay, and for this one, here in our own country, has one of the world experts in this business of the microRNA. So she is still well respected, renowned, the authority of this microRNA field. And another type is SNRNA, abbreviated in SNRNA, and it stands for small nuclear RNA. In the nucleus, some small RNA also do exist, and it is also non-coding RNA. Yes, but actually its function is also involved in protein production, kind of indirectly by its job is intron removal. What's intron? We are going to get into that later in this chapter. And so briefly put, introns are some of the in the middle interfering segment of the messenger RNA area, and to complete the RNA production and the following subsequent protein production in eukaryote, you should remove all those segments of intron correctly, and the machinery responsible for this, the removing the intron area, as you can see over here. This is one gene, and this intron area removed, and the yellow area will be joined together to finally have one mature RNA, in this case as messenger RNA. Although introns also present in tRNA sequences too, but always try to stay within the boundary of the standard information. So usually it does only it is pertinent only to the case of a messenger RNA. So important thing is there is another type of RNA which is non-coding, but its function is in removing the segment of messenger RNA called an intron, is probably. And another very similar in pronunciation, SNRNA, small nucleolar. So nucleus is the whole nucleus where your DNA is present, but there is another certain segment on DNA. It's called nucleolus, and this nucleolus is the area where ribosomal RNA. There are several versions of the ribosomal RNA. So ribosomal RNA's production. What do you mean ribosomal production? Transcription of ribosomal RNA. In other words, these areas, the ribosomal RNAs are present together in cluster. So example, like in cluster. Ribosomal RNA usually they are not spread over whole entire DNA on the chromosome here and there, like dispersed, but they are tightly, like they present in the same location together. So those areas, so that it's convenient to have this ribosomal production at the same time, and also assemble ribosome subunit, small and large subunits in order to assemble each subunit, what it takes is many different ribosomal protein plus ribosomal RNA, and you must combine them together. So this assemble ribosome subunit is another function. So one is ribosomal RNA production, and second is ribosome subunit assembly. And so during that procedure, during the ribosomal production, this ribosomal RNA needs to go through a further tailoring reaction. We call that as a simply edit the ribosomal RNA, which is carried out by another RNA edit RNA, and those RNAs are called small nuclear RNA, SNRNA abbreviated. Okay, so those are that. So those area that types of RNA that was just mentioned are all produced from the area of genes on the genome. But on the contrary, majority of the area of the DNA are not at all involved in RNA making activities at all. That means those areas are not the gene, right? And one interesting association, relatedness between those non-gene area and its chromosomal structure in terms of its condensation is this region is called heterochromatin. You know what the chromatin is, right? Chromatin is like a relatively loosely condensed chromosome structure, and chromosomes are only can be found in just right before the cell division, onset of the cell division. So most of the other period of a cell, like a lifespan, your DNA will exist in the form of a chromatin, right? Having still at the stage of chromatin, you do have this histone DNA plus wrapped, histone protein, and around which DNAs are wrapped. So this so-called nucleosome structure still exists, but only the difference is this in the chromatin as compared with the chromosome, the distance among each individual unit of nucleosomes are far away than it does in the chromosome. But these heterochromatins are usually, this heterochromatin can be found very prominently and exclusively in 3 different areas of a chromosome. If you divide this whole, let's say this is the entire your chromosome, and this area and the other 2, the edge of linear eukaryotic chromosome, we call them as a telomere, whereas the central area of the chromosome is called centromere. And these 3 areas, telomere, 2 telomeres, and 1 centromeres, if you look at the chromatin organization, they are in terms of a heterochromatin, even though that's the chromatin, there are 2 different type of chromatin. One is called hetero, chromatin. Maybe it sounds like something different chromatin. And another type of chromatin is euchromatin. And the difference is like heterochromatin is more condensed. And euchromatin is more relaxed. So even though they are both of them are chromatin, but their degree of condensation is somewhat different. This heterochromatin area, and I'm not saying that the heterochromatin areas are only can be found on either in telomere region, at the edge of the end of a chromosome, or the central area of a chromosome, which we call as centromere. But mostly speaking, and also characteristically speaking, if you look up the chromosomes on this telomere as well as centromere, they are like almost always they are in like heterochromatin structure, which is more highly condensed. And those areas, another characteristic is their DNA sequence area. I mean, their DNA sequence in the heterochromatin area, which is like, for example, like there are the telomeres and centromeres are something that constitutive heterochromatin. Meaning that they are always 24/7 exist in the heterochromatin structure, almost always. So which means telomeres, another telomeres at the bottom, and centromere area. These three areas are such example of a constitutive heterochromatin. And what's more important about this thing is those constitutive heterochromatin areas are their DNA sequence. Is highly variable among different individuals, for example in human. Like if you try to think about what's the average DNA sequence difference between two randomly picked human individual, if they were compared like their DNA sequence, how much difference do you expect? Something like 99.9% will be identical, right? So here only we are talking about only about 0.1% difference in their DNA sequence. But this DNA sequence, all these DNA sequence differences are a result of, once again, the mutation, right? Which occurred randomly in each different individual. So that's the amount of DNA sequence variation you can expect out of any randomly picked two human individual. However, here the point is those differences are not something that equally, although here is something mysterious thing, although the mutation occurs randomly. So if that is the case, why only those DNA sequence variation can be more frequently found in only these three areas where we define as constitutive heterochromatin area. Why is that so? The answer is these areas, this so-called constitutive heterochromatin areas, don't have genes. They don't have any genes? Well, almost. So, and that's the reason why this so-called constitutive heterochromatin areas contains much higher the variation in terms of DNA sequence. Can you understand what's the kind of a correlationship? Why would the heterochromatin area have higher DNA sequence variation than other area containing many genes? It's a kind of a result of the natural selection too. Although mutation occurs randomly, so that means mutation can hit. They don't care whether you are the area they hit the mutation is a part of the important gene or some junky area where there's no genes are present. The mutation will occur randomly. However, those individuals who happen to receive this mutation on their important gene sequence will have much more difficulty in a proper way of life, in other words, in survival or producing more number of offspring, because usually most mutations are bad one, deleterious, maybe spelling. So in other words, having such a bad influence on the health of the individual. Only very, very small fortunate event where the consequence of the mutation can actually provide the new function or ability which can help or the beneficial can be beneficial to the individual. Most of them are simply will produce bad effect on the function of this gene. So those individuals who happen to have this mutation on the gene area probably would have been eliminated, okay, throughout the course of this evolution by natural selection. But the mutation occurring on this non-gene area, where those heterochromatin area, somehow there is a really high correlation with where those heterochromatin area does not contain any genes. So it's okay for them. Doesn't affect. Yeah, if you have any kind of DNA sequence alteration, so be it, because I don't care, because I'm not going to suffer from the consequence. And that kind of a difference is accumulated over a long period of time, and now the discovery is like the observed consequences that, wow, you have such a higher DNA sequence variation in those heterochromatin area, whereas if you try to pull out the hemoglobin DNA sequence hemoglobin, the gene sequence, and then try to compare the DNA sequence with many different individuals. You wouldn't be able to find any DNA sequence changes among different individuals. If that is the case, then the mutated hemoglobin will probably have a difficulty in normal function, so that individual will be in trouble. So the reason why I'm going over this kind of the heterochromatin and their high DNA sequence variability is because I have some agenda. Yeah. Because of that, the heterochromatin area is a like easy choice and preferred popular choice for the practice of a new technique like this DNA profiling. DNA profiling is try to identify this DNA sequence variation among each different individual. So like DNA profiling, another name for calling this DNA profiling is DNA fingerprinting. Like your DNA sequence can be utilized as if like your the fingerprint to represent your true identity. If you use the DNA sequence area on like the gene heavy area, like where you have many important genes, you cannot make use of those DNA sequences to prove of your true identity, because everybody else will be identical in those areas of the DNA sequences. But in this heterochromatin area, each individual DNA sequence is exclusive and unique. So that is such a very good item to use in such purposes. This DNA profiling area, in particular, in applying those heterochromatin area in DNA profiling activity, particularly two DNA sequence elements has been traditionally has been very useful. So, for example, one segment of such DNA is called satellite DNAs, and the other one is called VNTR. You don't really have to know exactly what they are, but the common characteristic of whatever they are is the repeat. DNA sequence repeat. So these DNAs in heterochromatin area, they don't have any meaningful genes, but usually contain such mysterious sequence unit of repeated sequences. For example, regardless whether it's a satellite or VNTR, the length or the kind of a sequence motifs may be different, but basically, for example, if AGA-CCT, for example, if that is the one such a unit repeat, and this AGA-CCT sequence appear in tandem in many repeat unit, like N, something 20 repeats or 40 repeats, something 30 repeats. Why they appear in such a meaningless repeats over there? And more importantly, the number of these repeats. Number of these repeats varies among different individuals. That's the key of using this area in DNA profiling. You may actually have this question like, why do we have such meaningless whole garbage area? It's non-essential DNA, does not function any. the gene, still we keep them, like based upon the economy and the efficiency of this, the process of evolution by natural selection. Those individual carries this non-essential DNA should have been eliminated long time ago in the first place. Why do we have everybody has this such non-essential DNA area? Straightforward answer is nobody knows. Still, so this should be the area of further research until we can have a clear idea about what's going on over there. So those are first. It's been already 1 hour, and I try to actually keep in the like a set limit of lecture length time wise. So actually I have only just begun to introduce this idea of DNA profiling, and I have to stop over here, unfortunately. So at least for now I will stop the lecture now and then continue on the actual technical aspect and the idea of DNA profiling, and the next week to to comply with some of the students' concern about my lecture is too long. Okay, that's what I'm going to do right now. However, let me actually say only this before I just call it a class. If this was instead of a university lecture to which your interest only seems to be about getting a good grade, let's imagine, and if this was some kind of a private lecture course provided by private institute like Hagwon, yeah, that's possible. Then would you be so upset that just because some of the lecture length is longer than what is supposed to be? I don't really believe so, because the more you learn, usually it's supposed to actually go to more of your benefit, isn't it? Because after all, you are the one who pays for whatever the cost of this the service. Okay. Yeah, there's a reason why my lecture became always a little bit longer than the what is supposed to be. Obviously, you know that like there are so much to cover, but there are so little time in terms of its frame. So this one benefit of this having this online lecture instead of having this face-to-face or person-to-face classroom environment lecture is that I can somehow a little bit of a like play of flexibility to extend the lecture period. So I was able to enjoy a little some of them, and still personally believe that that is something that probably that's something that you shouldn't be so unhappy about it, but it's only me, it's my opinion. And if I were in your side of the shoes, probably I admit I would think differently. So having said, I hope you can continue enjoying the rest of the weekend. And once again, sorry about being a little bit late about this lecture recording and uploading due to my personal kind of situation. So bye bye.